Let's look at the human blood groups. We'll need to understand some terminology first. Proteins on the surface of the red blood cells, called antigens or agglutinogens, determine your blood type. Antibodies are proteins located in the plasma that will be used against blood types you do not have, foreign blood types. And these are also called agglutinins. Foreign antigens can cause clumping, and this is called agglutination, where red blood cells are brought together into clumps, allowing your immune cells to attack them. The antigens A and B bring about the greatest transfusion reactions, so let's look at the ABO blood groups. The ABO blood groups are based on the presence or the absence of two antigens or agglutinogens, and these are antigens A and B. There is no O antigen. If you are type A, you have antigen A on your surface. If you are type B, your red blood cells have antigen B on their surface. If you are type AB, you have a combination of both A and B antigens on your red blood cell surfaces. If you have type O blood, you don't have either A or B, so you don't have any of these antigens on the surface of your red blood cells. Your blood type is genetically determined. Let's look at the antibodies. You will have antibodies in your plasma for the antigens you do not have. In other words, for foreign antigens. These antibodies appear in your plasma shortly after birth. They are called agglutinins. If you are type A, you will have anti-B antibodies. If you are blood type B, you will have anti-A antibodies. Remember, you will have the antibodies for the antigens you do not have. If you are type AB, since you have both antigens, you will not have any antibodies. If you are type O, since you do not have any of the antigens A or B, you will have antibodies for both anti-A and anti-B. Rh blood groups involve the presence or the absence of an Rh antigen called agglutinogen D or Rh factor. If you have the Rh factor antigen on your red blood cell surfaces, you are Rh positive. If you do not have the Rh factor, you are Rh negative. Most Americans are Rh positive. This table lists the 8 major blood types. It assumes prior exposure to Rh antigens for Rh negative individuals. Recall that Rh negative individuals will not have Rh antibodies until after exposure. For each blood type, the antigens and antibodies are listed. In addition, important transfusion information is given at the far right. This table tells you what blood types this particular blood type can receive from and to what blood types can they donate. Stop the video and look at this table. You may want to copy it. I will be asking you information based on this table on the exam. The most common blood type in the US is O positive, and the least common is AB negative. The universal donor blood type will be a blood type that can be given to all of the different blood types. The universal donor is O negative. This is because they have no antigens. A antigens and B antigens, as well as Rh factor, are all absent. Hemolytic disease of the newborn, or erythroblastosis fetalis, may occur if the mother is Rh negative and the fetus is Rh positive. If this is the mother's first birth, she will come in contact with the Rh positive blood of the baby and produce antibodies. Rh factor antibodies are not present until after exposure. After this initial exposure, the mother will have antibodies which can attack the fetal blood cells if she has another fetus that is Rh positive. To prevent this, pregnant women who are Rh negative are given 2 shots of RhoGAM, one during their pregnancy and one immediately after delivery. This prevents them from producing the antibodies for Rh positive blood, which will protect future pregnancies with Rh positive fetuses. When mismatched blood is infused, a transfusion reaction occurs in which the recipient's plasma antibodies attack the donor's red blood cells. By binding to the foreign antigens, causing agglutination, immune cells then can attack the foreign red blood cells, causing them to lyse. This will release hemoglobin. If an individual is to receive a blood transfusion or an organ transplant, it is important to know the blood type of the donor to ensure that the blood type does not cause a transfusion reaction within the recipient. Let's look at how blood typing is done. Blood samples are mixed with anti-A, anti-B, and anti-D or Rh factor serums. Coagulation or no coagulation leads to determining the blood type. This is an example of a plate that we use in lab. There are 3 wells. The wells are labeled A, B, and Rh factor. In the well A, you will put an anti-A serum. The B well will have an anti-B serum, and the Rh factor well will have an anti-Rh factor serum. These are represented by the blue, the yellow, and the black, which is actually clear in the lab. A drop of the unknown blood will be put into each one of the wells, and the contents of the wells will be mixed. You will then wait to look for agglutination or clumping. If agglutination occurs, that means that the blood has the antigen of the same type as the antiserum. Let's look at an example of an unknown blood type. After we follow the procedure and wait for agglutination, in this sample we notice agglutination occurring with anti-A serum. There is a lack of agglutination with anti-B or Rh factor, anti-Rh factor serum. What this tells me is that the antigen A is present, but this blood type does not have antigen B or Rh factor. The results then would be A negative. This is a second sample. Agglutination occurs with anti-A and anti-B, but not with anti-Rh serum. That tells me that I have antigen A and B on the red blood cell, but not Rh factor. This blood type then is AB negative. In this sample, agglutination occurs only with anti-B serum. That tells me that that is the only antigen present on this blood sample. Therefore, this individual is B negative. I want you to stop the video and see if you can figure out this blood type. It is O positive, since the only antigen present is the antigen for Rh factor. In this slide, I have 6 samples of blood with the blood typing results. Stop the video and try to determine the blood type for each one of these samples. Sample 1 is AB positive. Sample 2 is A positive. 3 is O negative. 4 is B negative. 5 is AB negative. And 6 is B positive. Before giving an individual a blood transfusion or a donor organ, you need to do a cross-match. Cross-matching involves mixing the donor's red blood cells with the recipient's serum and looking for agglutination, and doing the opposite as well. If agglutination occurs, a transfusion reaction has happened, and that recipient should not receive the donor blood. Serum is plasma without the clotting factors. This is a flow chart of a cross-match. On the left, we'll begin with the patient's serum and the donor's red blood cells. These samples are mixed. If agglutination occurs after the mixing, the samples are incompatible and a transfusion reaction would occur. This is what's happening on the lower right. If no agglutination occurs, then that means that you can proceed with the transfusion because the blood samples are compatible. What else do you need to know? You need to know what blood types are compatible. In other words, when they are mixed, they do not cause a transfusion reaction or agglutination. For example, I could tell you that a patient has a certain blood type and they need a transfusion. What type of blood then can they receive? For example, if patient A needs a blood transfusion, given patient A is type AB positive, what blood types can patient A receive? Or I could also ask you something such as, if Sally wants to donate blood, what blood types can receive her blood? You will need to be able to identify the formed elements on histology slides, the erythrocytes, thrombocytes, and the different types of leukocytes. Know the normal ranges for the erythrocytes, the platelets, and the total white blood cell count. In addition, know the functions of each one of the formed elements. Use your table 17.2 in the textbook for the normal ranges of the different formed elements. I will have some assignments for you in mastering. Those assignments will have histology slides that will help you identify or learn how to identify the different formed elements and to test yourself as to whether you do know the formed elements or not. In addition, PAL in mastering, PAL, will have images of histology slides. I encourage you to go and look at those histology slides. This concludes the videos for Lab 1.